I'm merely doing what you asked. Any questions on stuff from Monday? Yeah.
Are we gonna start at right here?
Yeah.
Okay.
That's where we ended. I mean, we
Well, we
ended.
kinda like quickly went through.
We ended there, but we can go wherever you want. What do you want to go back to?
No, I just the next 3 slides that we briefly discussed. Alright, let's rediscuss them.
Okay, sure, we can do that. Questions about lab yesterday. We spent a ton of time in lab. If you have questions, I've already seen 3 of you, I think, since then about the R script. Yeah.
I have one more question about that. The factors that affect functional responses that we went over on the board, like handling time and the other 2, are those different from the types of functional responses?
Yeah, so those are the underlying factors that lead to functional responses taking on different forms.
Okay.
So the reason that as you increase density, the number of prey killed and captured goes up is because as density goes from 0 to a lot, you should expect the number of prey killed and captured to go from 0 to a lot, right? It just makes sense. There's more prey. You should capture more prey. Except that you can only capture as many prey as you can catch. Now, if prey density is really low, you're catching way fewer prey than you could be capturing, and so handling time isn't really constraining you because you're encountering prey so seldom. So you capture prey and you handle it, that handling time isn't getting in the way of you finding the next prey item because it's going to be some time before you find the next prey item. So this curve begins to increase at a steep rate, at low densities, but as you get to higher and higher densities, you're capturing more and more prey. With each of those prey, there is some handling time. So now finally there are enough prey that handling time becomes something of a constraint, which is why this curve, it increases, but it increases at a decreasing rate. So as you get to higher and higher densities, the slope of the tangent line is shallower and shallower. That's the rate at which you're gaining prey. So this looks like a diminishing returns curve. It's not really a diminishing returns curve. There's still plenty of prey because you guys were always replacing the discs. So prey density was staying constant. So it's not that you're slowly depleting that patch. It's that you now have enough prey that those short little handling times is beginning to cut into the total amount of time that you have for searching for new prey. So this has this shape because of a handling time constraint. In theory, when people were picking up the discs for you, you were freed up from that, in which case, this should just continue going straight. The number of prey that you capture should be directly proportional to the density because you don't really have any handling time constraints. Now that's actually not how your curves turned out, possibly for a different reason, which may be that you had trouble because you were going so fast. You have trouble distinguishing when you were on a prey on a disc versus when you weren't on a disc, especially at these high levels, because everything on the table was discs in a sense. So the things that we talk about are the underlying characteristics of predator and prey that lead to these things being different shapes, these curves being different shapes.
So the different shapes are the types.
Yep, the different shapes are the types. This is a type 2, this is a type 1, and we attempted unsuccessfully to mimic a type 3. A type 3 is an S-shaped curve going up to some asymptote. So this asymptotes out also at a point where you're just satiated. You can't handle any more than you've already handled. The type 3 is a situation where you either have to learn how to handle prey, and you learn better the more often you come into contact with prey, or you switch prey items such that you focus on one type of prey item at high densities and another type of prey item at low densities. So a prey item has to be present in the environment frequently enough that you will go and capture that prey, and it has to be at a certain density before you'll mess with it, because there's something else in the environment that is at a higher density that you're foraging on.
And which experiment did we do that tried to mimic that?
So the other thing that happens with this is when you have prey that are in patches, because when prey are in patches, you also need to be learning what the patch dynamic is. So are the patches that I'm coming into contact with, are they large patches or are they small patches? You know that when you guys were foraging, if you were watching one another, if you had the discs that were in little patches, and at my table that I was at, I sometimes did this to you. I had big patches, I had little teensy-weensy patches, and then I had patches that were intermediate of size. What this does is it throws uncertainty into it, so that if you happen to start off in this patch, you collect one, oh, then you find another one, then you find another one, then 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 another one, and again, and again, and again, until you've depleted that patch, until you stop finding them. Then you move and you find yourself in this patch. You might collect all 3 of them, but then you think that there are more there because you just got out of a patch that had a lot. It takes you a little while to learn that patches are variable. And so oftentimes what happens is at low prey densities, you're learning about patch quality, which is a type of learning, but it's a learning that is due to variation in patch quality. If all of your patches were roughly the same size, that learning curve is faster. My guess is that most of you probably arranged your discs in ways that the patch sizes were not terribly variable at low densities. For Claire's, I think we put 2 patches with 2 in each one. And in the case of you foraging, if I recall correctly, I put all 4 discs in one patch. And you just wandered around and didn't find anything. And then, knowing that, when you got to 8, we didn't put 2 patches, one with 4 and one with 4. We put, what did we do, Claire? I was messing with patch size with the 2 of you. We didn't do 1 with 8, but we messed with it. We might have done like 4, 2, and 2 or something like that. So anytime you have this uncertainty, it takes a little while to get a feel for what is the average patch size. That then determines kind of how long you should stay on average in any given patch. It isn't the quality of the patch itself. It's actually the average quality of the patch for the environment that you happen to find yourself in. So the type 3 scenario in your data doesn't really look like a type 3 scenario, and it's probably because the rest of you weren't monkeying with patch size the way I was monkeying with patch size with Claire and Alexis. Also, there's a fundamental difference in the way you had to handle prey and the way Hulling's students had to handle his prey with the way he set up the tables that they were working on. Other questions about that? Okay. So we were talking about snakes and newts. The snake, Thermophis cercalis, is a predator on the newt, Tufa granulosa. The newt has a toxin, tetrodotoxin, which is a sodium channel blocker, and the snake has evolved resistance to tetrodotoxin, and it's in this paper that we start getting into the molecular mechanism by which this occurs. We saw in previous work that there is a trade-off such that those snakes that are resistant to the tetrodotoxin tend to be slower under normal conditions. Those individuals that are faster under normal conditions are more susceptible to the effects of tetrodotoxin. So then the question is, how is this actually being accomplished? It turns out that if you look at the sodium channel, which is a transmembrane protein, at the outer pore of domain 4, there are a series of mutations, and these mutations have been compared to the relative resistances of different snake populations. Thermophis atratus, Thermophis couchi, and Thermophis tertalis all have really high resistance, and they all have well, they don't all have, but in 2 cases, there is a mutation in this amino acid residue, so I don't know how many, there are over 1,500 residues in this, amino acid residues in this sodium channel, and this one happens to be at 1568. There's a mutation here, and for Thamnophis calci, there's a mutation here that is unique that none of the other lineages share. They give it not quite as much resistance as Thamnophis totalis and Thamnophis intratus, but pretty close. So there are other mutations in the sodium channel, but these mutations don't have as big of an effect on the function of the sodium channel in terms of its ability to function when in the presence of tetrodotoxin. Now, not surprising that it occurs at the portions of the amino acid sequence where these things are on the outside of the cell membrane, because that's where tetrodotoxin is going to be interacting with the sodium channel. In this paper, what they did was they concentrated on 4 populations. 1 population that is allopatric with the newt, 3 populations that are sympatric with the newt, low resistance, medium resistance, and high resistance. And they looked at the particular amino acid sequences that are changed. And in this case, this is from a paper with Jansen and Brody where they looked at the sodium channels in a wide range of snakes. This was published after this paper was published, so they were just looking at the Willow Creek snakes, and the Willow Creek snakes at this time were the only ones known to have this particular change in the amino acid sequence of the sodium channel. What they did was they cloned the DNA. They took the DNA of a human, they attached the last little bit at the end here from the 4 different snake populations to human DNA. So if you have a DNA sequence, that is the DNA sequence that specifies the sodium channel, most of this DNA sequence is from human DNA. We have sodium channels because we have nerves. And then the end of this, the domain 4 section, they took that and they did the snake DNA. But they did snake DNA for Willow Creek, for Benton, for Warrenton, and for Bear Lake. And so there are 4 different DNA sequences. Each of these would result in these different amino acid sequences in this domain 4 area around where they think the important mutations are. Why did they create this stuff, which is called chimeric DNA? You guys remember what a chimera is?
It's like it has like one animal's body and their own head or something.
Yeah, so the Sphinx in Egypt is a chimera because it has a human head on a lion body. A griffin is a lion's body with bird wings and a like hawk or eagle head. So it's these mythical creatures that have parts of different animals combined together in the same animal. Why would you use chimeric DNA? Because what they're going to do with this DNA is they inject this DNA into a frog egg. Then the frog, as it starts to form membranes, expresses the genes in this chimeric DNA, and so they will express whatever kind of sodium channel is coded for in this chimeric DNA. Why would you use a chimera type DNA rather than just put the entire snake sodium channel gene in there? Why would you go to the trouble of creating human snake chimeras?
To make sure that it's actually the sodium channels that are resistant to.
Well, they actually just did this to the sodium channel gene. So they cloned the sodium channel gene, but the sodium channel gene was made up of a human part and a snake part. They didn't do the whole genome. They just did this particular gene.
but within that gene, the bulk of the gene is human DNA, the rest of the gene is snake DNA. Do you have sensitive or, do you have tetrodotoxin sensitive or tetrodotoxin resistant sodium channels in you? Oh yeah, you're sensitive.
So it's to test the like different levels or different concentrations of tetrodotoxin resistance in each of the.
So that's why they use DNA from all the different snake populations. But that is only the last part. All the rest of this DNA was the same, and it was all human.
Whether that mutation itself is the resistant part.
If you use all snake DNA, maybe resistance is imparted by not just this stuff that's at the end in domain 4, but maybe it's something back in domain 1 that is responsible for imparting resistance. And so by taking a sensitive sodium channel DNA and have that be the bulk of the sodium channel gene, and only making this last little bit of domain 4 the part that you alter, it basically narrows in on that and says, okay, if these things differ, it is because of something that's happening in that section, because we know that the rest of that sodium channel is a human sodium channel, and as a result, it should be sensitive. Do you guys watch like crime shows like I don't know, Rizzoli and Isles, CSI, et cetera, et cetera? Have you ever seen people getting killed by tetrodotoxin in any of these shows?
No?
Oh, really? Oh, I have seen it numerous times. And back before this particular case study became well known, you didn't ever see this in cop shows, but now in like forensics TV programs and stuff, you see tetrodotoxin being worked into storylines on a fairly surprisingly regular basis nowadays. Used to, you could probably kill somebody with tetrodotoxin and not get caught, but now it's well enough known that you probably couldn't. Not that I advocate that you kill anyone. I'm not saying that anyone should kill anyone with tetrodotoxin or anything else.
Put that on video.
But it's becoming more commonplace in fictional storylines because it has a neurotoxic effect. It shuts down sodium channels, which shuts down nerve impulses, which then paralyzes you. Have any of you seen Jane the Virgin? Watch Jane the Virgin program? Anybody?
What
Really?
It's on
That
Netflix.
show.
It's on
Caleb's favorite TV
Caleb
show.
loves that show.
Watched it 3 times too.
What?
I already heard of that show.
I think it's a movie. Is it a movie or a show?
It's a TV show. It's a spoof of telenovelas, and in this telenovela, Jane the Virgin has married this guy named Michael. Michael got shot at their wedding. He recovered, but then he died mysteriously. They buried him, but there's this other character in the show who's like this evil criminal mastermind who hates Jane and all the people around Jane. He actually didn't die from complications of the gunshot. He was actually paralyzed by tetrodotoxin. She admits this like many, many episodes later. She dosed him with tetrodotoxin, which paralyzed him, made him look like he was dead, then went and dug him up and sent him to Montana because of, while he's knocked out in tetrodotoxin, she gave him electroshock treatments that basically wiped out his memories, and so now he goes by Jason instead of Michael because it's a telenovela, and these kinds of things happen in telenovelas. But so my girlfriend is making me watch Jane the Virgin because she got into it, and so we're now in the last season. And I was watching it the other night with her, and all of a sudden there's this criminal who's in jail admitting to one of the characters that she dosed Michael with tetrodotoxin. I'm like, and there's tetrodotoxin again. This is in season 5, if any of you are interested in. I think season 5, episode 1, if you want to see
You should watch
tetrodotoxin
it in class.
written into, we're not. Written into a fictional storyline on TV. So, by using snake DNA in combination with human DNA, it makes sure that you're isolating just that section of the snake DNA that you think is important. You put it inside of a frog egg. The frog egg basically uses that gene as that gene gets transcribed and translated, it produces this chimeric sodium channel from the chimeric DNA. And then, once they have that, they can basically use a technique called patch clamping. It's where you put a little glass syringe up against the membrane of a cell, and you can use that to measure the electrical charge on the cell. And then they dosed these eggs with tetrodotoxin and then gave them a little bit of an electrical charge to see if they would then open up their sodium channels, because these are voltage-gated sodium channels. And if they did, they would observe a change in membrane potential that would indicate that these sodium channels were opening up and letting sodium channels flow into the frog egg. And when they did this, they found that they could calculate what the ratio of unblocked to total sodium channels are in an egg, and they found that in this first panel, this is comparing, well, I have to remember myself, it's clones to, yeah, so it's easier to read on the thing. Channels with entirely snake sequence are now more resistant to TTX blocks than chimeric channels. So this is comparing channels that are total snake DNA to channels that are chimeric DNA, and if they're not resistant, their resistance to tetrodotoxin is much worse than if they are resistant. And so this is, I think it's whole snake and chimera, but they're not significantly different. And here the lines are so close that you can barely tell that there are two of them at all. And so they're just basically showing that the chimera performs similarly to the actual whole snake, which indicates that it's really that last portion of it. This then shows it for the different populations: Bear Lake, Warrenton, Benton, and Willow Creek. And you see that Willow Creek resistance is much higher compared to the other populations, and the Warrenton population isn't that much higher than a non-resistant channel.
So the first line is that just all human DNA as kind of control, or?
No, it's a snake that doesn't co-occur. Let me go back. These colors, the purple is Bear Lake, yellow is Warrenton, orangish is Benton, and red is Willow
And
Creek.
what are the differences? I know you said them at the beginning very quickly, but can you like say them again?
3.6 mass-adjusted mouse units. This is the amount of tetrodotoxin that blocks 50% of the channels. So it takes very little tetrodotoxin to block Bear Lake channels. It takes more to block Warrenton channels. It takes even more to block Benton, and it takes a ton to block the channels of Willow Creek. And so these are measures of resistance from least resistant to most resistant across these four populations. Yeah.
Will you go to the next slide, and what was that in the first graph, the purple lines?
So in the first graph, this is two purple lines.
Oh, that's two.
One of the purple lines is all snake DNA.
Okay.
The other purple line is human snake chimeric DNA.
Okay.
And they're testing to see if the chimeric DNA gives you any different of a response from the total snake DNA. Here is the same thing. In this case, it's for the Benton population.
So there were just two different locations. That's why.
This is Bear Lake, this is Benton.
Okay, got it.
Two lines, one the chimeric DNA, one whole snake DNA. So what's the genetic basis of the channels that are being cloned? Is it all snake DNA, or is it a human snake chimera? Once they know that the chimera performs at kind of the same level as whole snake, they just go on after that and do everything with chimeric DNA. So this is now Bear Lake, Warrenton, Benton, and Willow Creek, showing you that the populations that they've measured out in the field in terms of their resistance, when you clone these things into frog eggs, you see similar rankings of resistance in the cloned channels as you do in whole snake performance, which we'll see here in a minute. What is seed again? So in this case, they changed valine to isoleucine at position 1561. And so in this case, they have two lines, 1561, from valine to isoleucine, and what happens when you have valine is you get an increase in resistance as a result of just changing isoleucine to valine. And then this is a diagram basically showing how Bear Lake, without tetrodotoxin, achieves a full depolarization. When you put it in the presence of tetrodotoxin, that depolarization is shut down. Willow Creek, which is the red population, it basically shows that Willow Creek's tetrodotoxin affected depolarization is essentially the same as the full depolarization. If you're looking at this in the original article, you can blow this thing up, and you can see that there's a black line underneath the red line, but they're basically right on top of one another. Basically showing tetrodotoxin blocks nerve impulse generation, depolarization, and tetrodotoxin doesn't in the case at Willow Creek. The difference between sensitive sodium channels and resistant sodium channels. Yeah.
Could you remind me which populations live among the toxic newts and which ones don't?
Yes. Go back. This is from the previous paper. This is the first figure in the previous paper. This shows the distribution of newts, at least in Oregon, Washington, and southern Canada, all the way over into Idaho. The stippled area is where the newts occur. Bear Lake is a population of snakes over here in Idaho that is allopatric to the newt, so there's no reason why they should be resistant to tetrodotoxin because they never come into contact with the newt. This is the allopatric population from the previous study of Thamnophis circalis in Idaho. We had allopatric Thamnophis circalis versus sympatric Thamnophis circalis, and we had sympatric Thamnophis circalis versus sympatric organoides, newt eater, not newt eater. Could be a newt eater, but doesn't because newts don't live there. Okay, so we're talking about many of these populations over and over again. So the easiest way to think about it is these. Bear Lake is here. Benton is one of the populations in the next paper. As it turns out, Warrenton is down further south, and Willow Creek is actually in the San Francisco area.
But all three of those are sympathetic with the newts.
All of those are along the west coast of the United States, and the newt goes pretty far south into California. So, everybody clear on what this figure is about? Less resistant, a little more resistant, much more resistant, way resistant, which is reflected in the amount of tetrodotoxin that is necessary to reduce the speed of snakes to 50%. 730 Massachusetts units versus 34 versus 15 versus 3.6. So the last paper that you guys read in E&E about this particular case study was this one where they looked for phenotypic mismatches. And phenotypic mismatches can occur in one of two ways. They can occur where the newt is way more toxic than the most resistant snake can handle. What would be the other way in which there can be a phenotypic mismatch?
This other snake is way more resistant than the most toxic.
Under these conditions, what happens to a predator-prey arms race? Yeah, it's over. One or the other species has won. In this case, the newts have won, and in this case, the snakes have won. There is no longer a predator-prey arms race because this level of selection on both species is now asymmetric again. And so what these guys did, in this case, and why do I not have the full map? I don't know why I don't have the full map. They went and looked at a lot of different populations. I think it was something like 34 populations, extending from southern Canada all the way down. This is the San Francisco Bay Area, all the way down in kind of central California. The newts have a range that extends down the Coast Range and also in the Sierra Nevadas. The Central Valley in California is a mesic area, it's drier, and as a result, you don't get newt populations there, but they have diverged, and one branch has gone down the Sierra Nevadas, the other branch has gone down the Coast Mountain Range. In western Oregon and Washington, they're all continuous there until you get over the Sierra Nevada mountains into the more arid eastern parts of Washington. And what they did in all of these populations is they measured what the resistance of snakes was, and they measured how much tetrodotoxin newts were capable of producing. And so they produced a map that basically shows you where newts are really toxic. And so, for example, this is about where Bear Lake is. This is where 10 Mile Creek is, another population of toxic newts. And down here, this is where Willow Creek is, outside of San Francisco. So this is a measure of a map of newt toxicity that basically extrapolates between a bunch of population estimates of newt toxicity. And the way they measure newt toxicity is they basically took little patches of newt skin off of the back of a newt between its shoulder blades, and then put it in a solvent, tried to extract all of the solvents, tried to extract all the tetrodotoxin out of the newt, and then ran it through basically a GC mass spec to measure the quantity of toxin that was in there. And then this is the resistance of the predator, of the snake. And you see that in some cases, the toxicity of the newt matches up nicely with the resistance of the snake. But down here around California, around San Francisco, it looks like the newts are not nearly as toxic as the most resistant snakes. And so you can basically put these two maps together. Do you have a question before we go on? You can put these two maps together and basically calculate to what degree does the phenotype of the newt in terms of its toxicity match the phenotype of the snake in terms of its resistance. And what you get is you get hot spots and cold spots. And the hot spots for mismatch are places where, in this case, the snake is much more resistant than the most toxic newts. One area where there is a hot spot is this area around, once again, around San Francisco, where you still have pretty toxic newts, but you have way-resistant snakes. Places where the mismatch is closer are in red, and you see that those are those places where snake and newt populations were similar in terms of both toxicity and resistance. And so looking at these three maps, this plots what the phenotypic mismatch is. And it occurs in a small number of populations, the purple areas. But in all of these cases, the mismatch is such that the snakes are unaffected by newts. So you can look at, this is the middle 15%. Sorry, the middle portion of the distribution of both prey toxicity and snake resistance. So it goes from 15 to 85. This is the upper 85 of snakes being way more resistant than newts. This is the lower 15 of newts being way more toxic than snakes can handle. And in a lot of these populations, the predator-prey arms race is going just fine. You have newts that are fairly toxic dealing with snakes that are fairly resistant. As newt toxicity goes up, snake resistance also goes up. But then you have a bunch of populations down here, and you have these four populations up here. What's happening with these populations down here?
The snakes are winning.
The snakes aren't winning. Who else is not winning? The newts are not winning. Why?
Either one of them is,
What's that?
either one of them is causing selection on the other.
Neither one of them are causing selection on the other. Prey toxicity, newt toxicity, is really, really low,
And snake resistance is really, really low also. These are basically populations that haven't yet entered the predator-prey arms race. What's happening with these populations up here that are in the gray area?
The snakes are more resistant because the toxicity is lower than the like mean 50%.
The newts are fairly toxic, but the snakes are way more resistant than the most toxic newts in these areas. So these are snake populations that have escaped the predator-prey arms race because the snake has become more resistant than the most toxic newts. No arms race here because the snakes won. No arms race here because neither of the two populations are toxic enough or resistant enough to get an arms race started. Everything in the white here is where arms races are currently occurring, where phenotypes of the two species interacting are similar enough in terms of their abilities that they can get this escalating selection going on, this escalating evolution. Newts become more and more toxic, snakes become more and more resistant. Yeah?
A basic question, are the newts evolving such that they produce more toxin or a more concentrated toxin?
That's an excellent question. Actually, it turns out now that they think it's, they're producing more toxin, not more toxic toxin. There are different isoforms actually of tetrodotoxin, but they all perform in about the same way. And they, from chemistry perspectives, when people look at these things with mass spec and stuff like this, those isoforms aren't different enough from one another in terms of their physiological effect of which isoform is produced matters. It's more a dosage effect than it is a different kind of tetrodotoxin. But one of the things that is true about this whole thing is the newt has been a much harder nut to crack than the snake has. We don't have any idea how newts make this. In marine organisms that are commonly known to have tetrodotoxin, things like fugu, the pufferfish, and there's a species of octopus that produces tetrodotoxin. They don't produce the tetrodotoxin themselves. They have symbiotic relationships with bacteria that produce tetrodotoxin. And so they have an organism living inside of them that makes the tetrodotoxin. They've evolved resistance, and in the process of them evolving resistance, they themselves become toxic. And as a result, if they get eaten, then they're toxic to any predator. But they've done studies, and I might have some of these things in here, where they've looked for those types of bacteria in newts and not found them. They've also looked for those types of bacteria in frog species that produce tetrodotoxin-like toxin, sapotoxin, and they don't find those bacteria in frogs. The poison arrow frogs that are found throughout Latin America and down into South America, those are species that don't have bacteria, but they get their toxins by basically eating ants, taking the toxins that are in the venom glands in the abdomen of ants, and basically metabolizing those toxins into other toxins that they then use. So when you go to the zoo and you see poison arrow frogs in the zoo, those poison arrow frogs have been in the zoo long enough and been without ant food for long enough that those organisms aren't toxic anymore because they lack the dietary source of where their toxins come from. So, in all cases, it's the snakes that have escaped the predator-prey arms race. Newts have not yet escaped the predator-prey arms race. Do you guys recall what it is about the snake sodium channel that makes them capable of escaping this and evolving relatively easily super resistance to tetrodotoxin? Okay. The mutation that is involved is a mutation that occurs that basically takes asparagine, which is in residue 1568, and converts that into, it takes aspartic acid and converts it into asparagine. So at residue 1568, it converts this from aspartic acid into asparagine. So how can you know that? Well, you can go back to the original DNA here, and it turns out that when you look at amino acids, every amino acid has a letter associated with it. It turns out that D is aspartic acid and N is asparagine. And so when you look at this, that's the mutation. What kind of mutation is that? When you look actually at the codon. It's a substitution. What kind of substitution? See, we went through all that mutation stuff at the beginning of the semester, now you need it. What's that? So it's a single nucleotide polymorphism. So it's just changing a guanine into an adenine. What is it? It is a transition, it's not a transversion. What's the difference between a transition and a transversion?
Transversion goes from, like A or G to T or C.
Yeah, so it goes from purine to pyrimidine or pyrimidine to purine. Basically switches types. This is guanine to adenine, those are both purines. Awesome. Those are both purines. Easier to change out a purine for another purine than it is to change out a purine and replace it with pyrimidine. This is a transition mutation, and because it's the easier type of mutation to occur, 2/3 of all SNPs that people have ever found are transitions rather than transversions. So this change in functionality is due to a single nucleotide polymorphism, and it happens to be one of the easier polymorphisms to pull off because it's transition. Yeah?
But if that's the mutation that's happening at, what was it,
Willow Creek.
Willow Creek,
Mm-hmm.
and the arms race is still happening there, what
It's
hap-
not.
Okay,
That's, Willow Creek is one of the soil populations.
so all of the purple areas on the map have that mutation.
Yep. There's one of these is Willow Creek, and I don't know which one it is, but one of these up here is it.
So what is creating the purple and blue areas on the map?
The purple areas are areas where there's phenotypic mismatch, but there's a phenotypic mismatch because both of them are so weak that nothing is happening. There are other parts, well, let me see. This is actually, the purple here actually has nothing to do with the mismatch. The purple here has to do with, does it? Now, it'd be nice if I had put figure caption in here, wouldn't it? I'm gonna have to go back and look at what the purple is.
Okay.
But so in theory, you would think that the purple and the yellow and everything would correspond to the phenotypic mismatch, but it can't because these are all really non-resistant snakes and really non-toxic newts. These are the really resistant snakes in the presence of pretty toxic newts.
Could it be their locations?
Yeah, but the locations would be easy enough to map. Let's just go and find.
It could be the locations of it because some of them are allopatric, Willow Creek for instance. So you couldn't really measure newt toxicity if the newts aren't there.
Well, Willow Creek is definitely in the presence of newts.
Barely, because it's not much.
Now, why am I looking for it in a server?
Hockey player?
I need to be looking. Let's go and find the paper. So these are the, I don't know why I don't have this in this particular version of the thing, but basically, the squares are predator resistance, the circles are prey toxicity. When we're down here,
here's Willow Creek, once again down here in the San Francisco Bay Area. What you have is really, really resistant snakes with only intermediate resistant newts. So this shows you all of the populations that they surveyed. They don't have Bear Creek in this study because there's no point in measuring Bear Creek because newts don't occur there. This is a population of introduced newts that probably got introduced from somewhere over in Oregon or Washington or something. So there are a couple of populations of introduced newts here. In those cases, both the snakes are not very resistant and also the newts are not very toxic, but that's an introduced population. So let's go find that last figure.
I think it says 28 localities, and I think there are 28 circles.
Okay, 20 localities. They're colorized by average newt toxicity. That's the reason that the colors don't make sense. So these are average newt toxicity. So the newts here could be higher toxicity because these populations of newts are higher toxicity. They're not, and that's why these snakes have way more resistance than the newts have toxicity because these newts haven't achieved this level of toxicity yet, and so the snakes have literally outrun the newts evolutionarily. Yeah.
I'm going to try to phrase this clearly.
Yeah.
How is it that a discrete set of potential mutations, primarily this SNP that we just discussed, leads to a sliding scale of resistance in the snakes?
So it isn't so much a sliding scale of resistance. What do you notice about the x-axis here? Always pay attention to x-axis. Point 0, 1 to point 1, that's the same distance as point 1 to 1. That's the same distance as 1 to 10, which is the same distance as 10 to 100. What kind of scale are we looking at? Yeah, it's a log base 10 scale. So these aren't continuous variations in toxicity. These snakes, or resistance rather, these snakes are basically 100 times more resistant than these snakes here because this is on a log scale.
All the points don't fall like right above 0.1, 1, 10, 100, like there's.
No, but the jumps, if you were to put this on a non-log scale, these in this cluster would be here, these would be 10 times away, and these would be 100 times away from these. Because it's a log scale. So what looks like continuous variation is actually big jumps. In the same way that this is the amount of tetrodotoxin needed to reduce Bear Lake populations to 50% of their baseline speed. Only takes 3.6 units of tetrodotoxin. At Warrenton, to reduce Warrenton snakes to 50% of their speed, it takes 15 units of tetrodotoxin. At Benton, to reduce them to 50% of their speed, it takes 34. At Willow Creek, to reduce Willow Creek snakes to 50% of their baseline locomotion, it takes 730 units.
Yeah, but those
This is
four
not
numbers
con-
aren't like
Hmm?
easily divisible by each other. Like what I'm saying is, are there discrete levels of resistance, or can you be at a level 1 or a level 1.5 or a level 2 or a level 2.5 or
So-
et cetera?
It's not exactly discrete, but it's certainly not continuous because there are these big jumps. Going from having these two mutants, so going from having this version, isoleucine, to valine, bumps you up. Now, all the rest of these populations have valine. So having this mutation, which I don't remember what it is, gives you a little more resistant resistance up to this level, but having this single one takes you from being so-so resistant to super duper resistant. So it's really more of a step function at that point. Does that make sense?
Yes.
It's not continuous in the way that human height is continuous. It's much more of a discrete thing, going from medium resistance to supreme. Supreme get you out of the arms race sort of resistance.
Right.
So, snakes and newts in the arms race haven't entered the arms race yet, have escaped the arms race mainly because the snakes have won. And the snakes won because the mutation is relatively easy to achieve because it's just a single nucleotide polymorphism. But the secret to understanding that is to look at the actual structure of the amino acids, because what we're basically doing is we're going from this to that. And what you have in this case is you have a negatively charged amino acid to a potentially positively charged, or at best neutrally charged, amino acid. So it changes the charge on the amino acid, and that amino acid sequence is out here at the outer pore, where tetrodotoxin is going to be attaching to the sodium channel. So when you change the charge on that puppy, that amino acid is going to be interacting with this thing. Tell me about the chemical nature of this molecule. Pardon? It's neutrally charged, except that it's not. So you have a positive charge here, you have a negative charge here, but you have to think about this thing as having three-dimensional structure. Just because the molecule itself has a neutral charge, there's a negative charge here and there's a positive charge here. Where else do we get charge nature in this molecule?
Partial negatives on the OH.
Oh my God, partial negatives on how many OH groups? Or partial positive charges and partial negative charges on all these OH groups? On all these OH groups, you have sticking off of the OH a partially positive hydrogen jutting out away from all three of the ring structures in this thing. You have a lot of positive charge in a tetrodotoxin molecule. When you have that positive charge in a tetrodotoxin molecule, what is it going to do to aspartic acid? It's going to suck right in there. All that partial positive charge combined with the negative charge on aspartic acid, it's going to bind there and it's going to stay there. As soon as you change it to either a neutral charge or a positive charge, which is what asparagine will have, then you get all these positive charges interacting with one another. What happens to tetrodotoxin? Way harder for it to bind. You have charges that are repelling one another rather than attracting one another. And so a single mutation leads to super resistance because of the molecular interactions of the things involved. The amino acid residues that are in the sodium channel combined with the nature of the toxin that is interacting. One gets in there, blocks the pore, and stays stuck there for long periods of time because it's stuck there through the charged nature of the amino acid residue and the molecule itself. And in the other case, it actually repels it. This is at the molecular level, what's happening when you get dosed with tetrodotoxin by a nemesis and a telenovela.
You die.
Well, yeah, you would die, or in this case, they've just paralyzed them. So you don't have to die from tetrodotoxin, but you certainly can get paralyzed by tetrodotoxin. You just generally don't want to mess with tetrodotoxin. You guys are all sensitive. We should not test that in lab.
Are all the,
Define for me what you mean by test that in lab, Jackson. Yeah, go ahead, Logan.
Jack is this-
Are all of these amino acid residues in, like, on the outside of the membrane? Are all of the
No,
ones shown here, or is
Um,
it specifically, I don't know, 1560 to 1570 that are on the outside?
All of these are, sorry, I'm having to flip back and forth. All of these here, from 1556 up to there, which is all the ones that are on the next figures, they're all in this section right here. So they're all on this portion that links subunit 5 to subunit 6, and those are all up here on the outside of the sodium channel. Here is 1550, 51, 52, 53, 54, 55, 56. So that's all that section. Questions about this?
About this picture?
No, it's the whole case
Oh,
study.
okay.
Yeah, look.
Probably thinking too far into this, but if you're doing this, mutation where it changes an originally negative charge, you have to do positive, wouldn't it affect the tertiary structure of the protein itself?
It would probably affect the tertiary structure of the
So
protein
could that-
itself.
Could that jeopardize its natural function as a sodium channel?
Sure, and what happens to snakes that are really resistant?
Get slow.
They're slower when they're not being challenged by tetrodotoxin. So the answer is, yeah, it probably does change the tertiary structure, which changes the ultimate function of that sodium channel under normal conditions. But that trade-off isn't big enough, as it's probably as important to be able to catch food that nobody else can eat in your environment as it is to avoid predation in this case. So yeah, that's a valid point. What other valid points are there to be made about this particular case study? In order for this to be a predator-prey arms race, there has to be a genetic basis for, in this case, the resistance of the predator. Because this involves a sodium channel, because that sodium channel is ultimately determined by a DNA sequence, sodium channels are heritable. What would it be nice to know about? We know the snakes are resistant. We know at the molecular level why the snakes are resistant, because it's a genetically encoded trait. We know that it must be subject to evolution because it's heritable and is subject to selection. We know which populations are resistant. We've calculated heritabilities on resistance in earlier papers, et cetera, et cetera. We know all this stuff about the snake. What do we not know? We know almost nothing about the newt. All we know about the newt is that toxicity varies from population to population. And we know that their eggs are toxic also, so females put tetrodotoxin into their eggs. We know that the tetrodotoxin is found mostly in skin and not in other tissues of the body, except actually in the liver and in the ovaries. Oh, and there's a recent paper that just came out a couple of months ago that shows that the tetrodotoxin, that the glandular structure of newts that produce tetrodotoxin in high amounts are structurally different than the skin glands of newts that don't produce high levels of tetrodotoxin. So there's actually a structural difference that is detectable under a microscope in the glands of those species. That came out just in the last 2 months. And that's it. That's all we know. We don't know anything about the genetic basis of tetrodotoxin production. They tried isolating the bacteria from newts using PCR markers that are found in similar bacteria that do produce tetrodotoxin. Didn't find any evidence that bacteria are responsible for tetrodotoxin production. So they think that tetrodotoxin production is endogenous, not due to an endosymbiont. But the newt side of things is more about stuff that we don't know than about stuff that we do know. Jack?
We might have already addressed this, but do the newts have the same mutation, or for them it probably wouldn't be a mutation, but the same difference in sodium channels that the snakes have that allow them to be resistant, because if you're producing something that's
There-
like super toxic.
There is a paper about mutant sodium channels that I have not read, but I'm assuming the answer to that question is yes, they do. Because they would just kill themselves if they didn't. So, and I just haven't gotten around to the full paper. Other questions?
What's this picture?
It's a picture of, oh, these are all the mutations. Oh, sorry. These are all the mutations. So there's a mutation from isoleucine to leucine, isoleucine to valine, glycine to alanine, aspartic acid to asparagine, glycine to valine. And in all of these cases, you go from neutral to neutral, neutral to neutral, neutral to neutral, neutral to charged, potentially, neutral to neutral. So the only one where you go from a negatively charged to either neutral or positively charged is the one that imparts super resistance. So it's just a kind of summary comparison chart. These are all the residues that are in this figure. 56, 61, 66, 68, 69. So it's just taking these letters, translating them into their amino acids by residue, and then showing you the structures of the amino acids. So that, once again, for me, an important thing for you to recognize is that we tend to silo knowledge in biology. If you're an ecologist, you think you don't need to know anything about organic chemistry or biochemistry. And if you're a biochemist, you certainly don't think you need to know anything about behavior. You couldn't understand this case study fully unless you had a knowledge of biology in a broad sense, knowledge of ecology, knowledge of behavior, knowledge of evolution, knowledge of some basic chemistry. It makes way more sense to you if you combine all those areas of knowledge than if you keep yourself in your little silos. Other questions? Your book talks about Crypsis in this chapter that is about predator-prey arms races. Would you expect Crypsis to involve the setup and maintenance of a predator-prey arms race? Why not? Alexis is shaking her head now, which is why I say why not.
Reciprocal selection.
Yeah, the possibility of reciprocal selection is much less so, because if you're more and more cryptic, that probably doesn't have a huge or very consistent consequence on the fitness level of the predator. And so it's hard to see how this would evolve into a predator-prey arms race, which involves prey becoming more and more cryptic and predators become more and more keenly sighted. If you are cryptic and you escape the notice of a predator, the predator just goes by and doesn't eat you, which works good for you, but probably doesn't have much of an effect on that predator's fitness, because it will just run across a less cryptic prey item and see it and eat it instead. And so Crypsis is an anti-predator mechanism that prey utilize, but it's hard to envision it becoming a predator-prey arms race. So the book then kind of departs from predator-prey arms races to talking more about normal predator-prey interactions that aren't as unusual as predator-prey arms races. And we're practically out of time, so we'll just stop there. Next time, we'll start with Crypsis and continue on through the remainder of the paper. Monday, we're going to read Jansen and Brody. That's Monday, right? That's when it's on the schedule. We probably won't be finished with the predator-prey chapter by then, but we will have at least gotten on Friday to the point where we talk about aposematism, which is what that paper is about. So we will have talked about that in class at least before we get down there. You have a question?
Yeah, what I think you're gonna answer in a couple days.
Ask it now.
Okay, I just know the difference between Crypsis and Aposematus.
So Crypsis, well, this would be good for you to know as you start to write your literature summary. Crypsis is when you have a coloration that basically keeps you hidden, allows you to camouflage yourself. And Crypsis can take on a number of different forms. You can be cryptic because you have a coloration that disrupts your body shape You can have crypsis because you look like anything inanimate in your environment. You can be cryptic because you color match your environment. You can be cryptic because you resemble a twig or a bird dropping or something like that. Aposematism is actually just the opposite of crypsis. It's coloration that calls attention to you, and it calls attention to you because you're harmful or toxic, and as a result, you become memorable in the mind of whatever predator just tried to eat you, so that they'll remember your color pattern and not try and go after you again. So they actually have two totally opposite effects. One effect is to go unnoticed, the other effect is to be super noticed. The problem in the paper that you guys are reading from Monday is that some people have argued that under some light conditions, the banding patterns of coral snakes might actually serve as disruptive coloration, a type of crypsis, not aposematism. So that's why they wanted to test between aposematism and crypsis. If you get a dark rainforest where the leaves are absorbing all of the red light, those red bands might not look like red bands all